Hi, in this lecture we shall study the structure of a generalized cell. The structure of a cell can be studied under a light microscope as well as an electron microscope. The modern technology enables us to isolate various components of cells including its organelles by a process of cell fractionation and study their structure and function in detail. During cell fractionation, the tissues are homogenized or disrupted with special instruments and the various parts of the cells are separated by density gradient centrifugation. This separation is achieved by spinning the homogenized or disrupted cells in a special medium in a centrifuge at medium speed. The various cellular parts separate out in different layers depending upon their size and weight, and density of the medium. Some cellular components require very high speeds for separation from other parts of the cells. This is achieved through ultracentrifugation. In the traditional system of classification, all organisms are divided into plants and animals. The cells of plants and animals can be distinguished by the presence or absence of the cell wall. Cells of animals and plants are complex and have a distinct nucleus and are called eukaryotic. On the other hand, the primitive type of cells, such as bacteria, lack a definite nucleus and are said to be prokaryotic. In prokaryotes, the nuclear material is directly submerged in the cytoplasm and is not separated from it by membranes. The eukaryotic cells vary greatly in size. They could be as big as an ostrich's egg. Most of the cells are microscopic and are not visible to the naked eye. Their size is measured in a micrometer. The use of modern technology has made it possible to study the components of the cell in detail. Plasma membrane or cell membrane is the outermost boundary of the cell. However, in most plant cells, it is covered by a cell wall. Cell membrane is chemically composed of lipids and proteins. 60 to 80% are proteins, while 20 to 40% are lipids. In addition, there is a small quantity of carbohydrates. Many biologists contributed to establish the structural organization of cell membrane. It was proposed earlier that cell membrane is composed of lipid bilayer sandwiched between inner and outer layers of protein. This basic structure is called the unit membrane and is present in all the cellular organelles. The modem technology has revealed that lipid bilayers are not sandwiched between two protein layers. The protein layers are not continuous and are not confined to the surface of the membrane, but are embedded in lipid layers in a mosaic manner. This discovery led to the proposal of fluid mosaic model. This model at present is the most accepted one. Cell membrane also contains charged pores through which movement of materials takes place, both by active and passive transport. Transport of materials is one of the vital roles it plays for the cell. It offers a barrier between the cell contents and their environment, allowing only selective substances to pass through it, thus it is known as differentially permeable or selectively permeable membrane. The substances which are lipid-soluble cross it more easily than others, therefore, it regulates the flow of materials and ions to maintain a definite gradient. Many small gas molecules, water, glucose etc. being neutral can easily cross, while ions, being charged particles, have some difficulty in crossing. Many substances which are not needed, constantly enter the cell by passive transport. Others are taken up against the concentration gradient, which means that they move from the area of low concentration to the area of high concentration. This uphill movement of materials requires energy and is termed as active transport. The energy used for this movement is provided by ATP. In many animal cells, the cell membrane helps to take in materials by infolding in the form of vacuoles. This type of intake is termed as endocytosis, which can be either phagocytosis, which means to engulf solid particles, or pinocytosis, which means to take in liquid material. In neurons or nerve cells, the cell membrane transmits nerve impulses from one part of the body to the other to keep coordination. The cell membrane helps maintain the cell's shape and structure, providing mechanical support and stability. Cell membrane protects the cell's internal contents from external damage, pathogens, and toxins. It also plays a role in cell signaling, allowing cells to communicate with each other through signaling molecules, such as hormones and neurotransmitters. Cell membrane helps in cell recognition, allowing cells to identify and interact with each other, which is important for processes like immune responses and cell adhesion.